let's practice working with Lac Operon genetics by doing some example problems. The map of the Lac Operon is POZY. The promoter the promoter region is the start site of transcription through the binding of the RNA polymerase molecule before actual mRNA production. Mutationally altered promoters P- cannot bind the RNA polymerase molecule. Predict whether beta-gal and permease will be expressed or not using plus and minus respectively. All right. The main addition in this problem from what we have seen before is the fact that you can have mutant promoters denoted P minus, which have lost the ability to allow polymerase to bind. And of course, if polymerase doesn't bind, then there is no transcription, leading to no expression of whatever gene, whatever allele happens to be linked to the mutant promoter. And of course, the first genotype is just the wild type Lac Operon, and that will have inducible expression, which means it will not be expressed when you don't have lactose or when you don't have IPTG, and it will be expressed when you do have or they will be expressed when you do have IPTG. Now, there are basically two ways to approach such problems. One way is to reason through the consequences of these genotypes, recalling the properties of these mutants, which one is dominant, which one is recessive, whether they act in cis or whether they act in trans, and so on and so forth. The other way is to draw out mechanistically what is going to happen, and we're going to use both approaches here. So the second mutant here is I plus P minus OC Z minus Y plus, and it's a partial diploid with I minus P plus OC Z plus and Y minus. Now we have a heterozygote between I plus and I minus, and that means there is repressor, wild type repressor available in the cell, and I plus is dominant over I minus, and it acts in trans. The other thing that we have is we have OC mutants, both of them, but since OC acts in cis, we only need to worry about the consequences of OC with regard to the alleles that are linked to OC or on the same DNA molecule as OC. And so let's get started. Let's look at this first allele, which is Z minus Y plus. And this allele will obviously not have any beta-gal expression because of the Z minus allele. So the only protein this allele can express is permease. However, we also have a P minus mutant promoter on this allele, and if you have a mutant promoter, polymerase cannot bind, and therefore no transcription will occur, and this allele cannot express any permease. The other allele cannot express any permease either because it's Y minus, and therefore we can write for permease minus in both conditions, since the first allele does not express permease due to a mutant promoter. The second allele does not express permease due to a mutant LAC-Y gene. Since the first allele is Z minus, to look at the expression of beta gal, we only need to look at the second allele. Now, this one has a functional promoter, but it does have a OC mutant which is constitutive, and OC is constitutive because it doesn't allow the repressor to bind, whether there is lactose or whether there isn't any lactose in the cell. And therefore, we will end up with constitutive expression of beta gal because of the OC mutant. Now, let's do the same genotype in a mechanistic manner. Now, I have a heterozygote between I plus and I minus. Since I do have one functional allele of LAC-I, I will make repressor in both the presence and the absence of lactose. However, in the presence of lactose, this repressor is going to undergo an allosteric transition because of the binding of allolactose or IPTG, giving me a repressor that is unable to bind the operator. Now, the operator is not a wild type operator but the mutant OC, and that means that the repressor is not going to be able to bind irrespective of whether you have lactose or you don't have lactose. And normally that leads to constitutive expression. However, we have a mutant promoter P minus on one of the molecules, so the chromosome, and therefore we don't have any polymerase on the bacterial chromosome, the promoter on the bacterial chromosome, whereas we do have elongating polymerase that binds to the other wild type promoter. And what that means is that there is no expression of Y plus in either condition, and that is what we wrote down with our reasoning. Similarly, we will have expression of beta gal in the absence of lactose because the repressor can't bind OC, and we will also have expression of beta gal in the presence of lactose, once again because the repressor can't bind the mutant operator, as well as the fact that the repressor has undergone an allosteric transition and is not able to bind even the wild type operator. And that's why we end up with constitutive expression for beta gal. Let's attempt the third genotype, which has a hit for the super repressor allele of the repressor with the I minus allele of the repressor. Let's do it both ways, so one by reasoning and the second by working it out mechanistically. we have wild type alleles of everything else, so we only need to worry about how a het between a super repressor and I minus would behave. Now we know that IS is dominant to I plus and I plus is dominant to I minus, with the implication that IS is going to be dominant over I minus, and this should behave like a super repressor. It makes sense because the I minus simply doesn't make a functional repressor, whereas the IS allele makes the super repressor, which represses or binds the operator irrespective of whether you have lactose or not. And since the I acts in trans, it doesn't matter whether I have IS here or here. And therefore, with this reasoning that IS is dominant and it acts in trans, we should get super repression, and so we should not get any expression of beta gal or permease whether we have IPTG or not. Now, if we work this out mechanistically, I minus will not make any functional protein, whereas IS will make the super repressor protein, and the super repressor protein will act in trans and bind the operator on both the chromosome as well as the F prime plasmid, both the alleles of the operator. It will bind to them and prevent transcription when you don't have any lactose present. Even when you do have lactose present, since this is the super repressor and it's the site where allolactose or IPTG binds has been mutated, so the super repressor will not undergo a conformational change and allosteric transition to prevent it from binding the operator. It will still bind the operator in the condition when you do have lactose, and prevent transcription, resulting in the constitutively repressed phenotype, as we reasoned previously. So far, we have discussed how the Lac Operon is regulated by the amount or the availability of lactose in the cell. As it turns out, the Lac Operon is also regulated by the availability of glucose, and it's not hard to see this from an efficiency perspective, since if the cell has access to both glucose and lactose, it would be beneficial for the cell to utilize glucose rather than lactose, since there is more of an ATP investment required in making beta gal and hydrolyzing lactose into galactose and glucose. The way glucose regulates the Lac Operon is referred to as catabolite repression. And normally, if you don't have glucose in the cell, then ATP, adenosine triphosphate, gets converted into cyclic adenosine monophosphate or cyclic AMP. However, glucose inhibits this conversion of ATP into cyclic AMP. Now, when there is very little glucose, cyclic AMP, there are high levels of cyclic AMP, which will bind this protein called the Catabolite activator protein, or CAP. And so cyclic AMP will form a complex with CAP. And what that allows CAP to do is to bind a DNA near the promoter of Lac Operon, resulting in a bending of the DNA near the promoter of the Lac Operon. And here's the -10 box, and here's the -35 box of the Lac Operon promoter. This bending of the DNA recruits polymerase, so it allows more and more polymerase molecules to arrive at the promoter and shoot off. The more polymerase molecules arrive and start transcribing, the greater the Lac Operon transcripts are made, and therefore the greater the number of protein molecules made. And therefore we end up with a picture of the following sort. So let's say we have glucose present and no lactose. If we don't have lactose, then repressor encoded by LAC-I will bind the operator and prevent transcription. Also, since there is glucose, the glucose will prevent the conversion of ATP into cyclic AMP, and therefore CAP will not be in complex with cyclic AMP and will not bind its binding site. And the net result of the repressor preventing the availability or the arrival of polymerase at the promoter, and the absence of a CAP cyclic AMP complex which recruits more polymerase, is that you will have no LAC mRNA. A second sort of situation that one might see is that both glucose and lactose are present. Now, since we have glucose, ATP will be inhibited from forming cyclic AMP, and therefore CAP will not bind its binding site and recruit polymerase. On the other hand, the allosteric transition of the repressor with lactose will prevent the repressor from binding the operator and allowing polymerase to bind to the promoter and carry out elongation to make LAC mRNA. And it will not be a lot of LAC mRNA since the polymerase is not being actively recruited to the promoter. And the last situation to consider is if there is no glucose available to the cell and the only source is lactose. Since there is no glucose, ATP can get converted to cyclic AMP, and there is a lot of cyclic AMP CAP complexes, and they will be able to bind the CAP binding site near the promoter and cause the DNA to bend and thereby recruit a lot of polymerase. And the picture may look something like this. Lots of polymerases will be running along the gene one after the other, making lots of transcripts, and that results in a very high expression level. Since we have lactose in the cell, due to the allosteric transition, the repressor will not be able to bind the operator and prevent the polymerase from binding to the promoter and transcribing Lac mRNA. And therefore, we here have two distinct mechanisms of gene regulation. And the first one that we have discussed extensively is when the repressor prevents transcription, and therefore it has a negative influence on transcription. So that's called negative gene regulation. The other mechanism we have is the catabolite activator protein CAP, which binds the promoter, bends the DNA, and recruits more polymerase, and therefore it has a positive influence on transcription. In other words, it activates transcription, and this kind of a protein is called an activator that has a positive influence. So a mechanism of positive gene regulation. Finally, let us do an example. The activity of the enzyme beta-galactosidase produced by wild-type cells grown in media supplemented with different carbon sources is measured. In relative units, the following levels of activity are found. With only glucose, there is 0 activity. With only lactose, there is 100, a level of 100, and with both lactose and glucose, there is a small amount of activity, a level of 1. And we are asked to predict the relative levels of beta-gal activity in cells grown under similar conditions when the cells are Lac-I minus and CRP minus. So let's start with drawing what is mechanistically going on in wild type. Here we have the Lac-I gene, the CAP binding site, promoter, operator, and Lac-Z. Now, when we have only glucose available to the cell, what that means is that CAP there is no cyclic AMP, and therefore CAP does not bind to the CAP binding site. The repressor is made by the Lac-I gene, and the repressor goes and binds the operator, since there is no lactose in the cell. And what that implies is that polymerase can't bind the promoter, resulting in an activity level of 0. Now, when we add lactose to the cell, that will cause an allosteric transition in the repressor, and prevent the repressor from binding the operator. Cap, since glucose is still there is very little cyclic AMP, and cap is not binding to the cap binding site. The lack of repressor binding to the operator means now the polymerase can bind the promoter and transcribe, just not at a very high level, and that's why we get an expression of 1 or activity of 1. And finally, when we have just lactose, then the presence of lactose causes the allosteric transition, and that means that there is no repressor preventing polymerase from arriving at the promoter. But in addition, we have lots of cyclic AMP. So this disk is cyclic AMP. Since glucose is not inhibiting the conversion of ATP into cyclic AMP, and that recruits a lot of polymerase to the promoter, leading to a very high activity level of 100. Now, what we want to do now is modify this picture in the two mutants, Lac I- and CRP-. So first, let's do Lac I-. Now, if we are Lac I-, that means we don't have any repressor protein. And what that implies in turn is that the picture does not change very much for these two conditions, since in the wild type there was a repressor, but the repressor was not able to bind the operator site due to the allosteric transition caused by allolactose. The picture will, however, change for the only glucose condition, since polymerase will be able to bind the promoter due to the lack of functional repressor, since we're looking at a Lac I- strain. And that implies that the glucose only condition becomes the same as the lactose and glucose condition, where cap doesn't bind cap binding site, but a repressor also doesn't bind the operator, allowing the polymerase to transcribe at a lower level, which means that in Lac I- you will get an activity level of 1. Next, let's look at the other mutant and what will happen in CRP-. But first, let's go back to the picture for wild type. Now, CRP is made by a different gene called CRP, and the wild type allele will be CRP plus. And if we change this to CRP-, it means we don't have any functional CRP in the cell, and therefore we can predict the levels of activity by simply erasing CRP from the picture over here. Now, this first condition is not going to change, since there is a repressor which is preventing the recruitment of polymerase to the promoter, and you will still have no transcription, since the repressor is preventing the polymerase from arriving at the promoter. Similarly, the second picture, where you had lactose and glucose, is also not going to change, since the presence of glucose implies that there is no cyclic AMP, or implied in the wild type that there was no cyclic AMP, and therefore cap could not bind the cap binding site. The only thing that's going to change in CRP minus is the last condition, where you have only lactose and no glucose, where in the wild type cap was bound at cap binding site, since there was no glucose to prevent the conversion of ATP into cyclic AMP, and therefore there was lots of cyclic AMP cap complex which was bound at the binding site and recruited lots of polymerase. Now, since we are in a CRP minus strain, there is no cap, and there is, even though you have a lot of cyclic AMP in the cell, the fact that we don't have any functional cap protein means that there will be nothing bound at cap binding site, and therefore nothing to bend the DNA and recruit polymerase, and therefore we will recruit polymerase at a slower rate, just as if you have glucose in the cell. And that means that this condition with just lactose will become the same as the condition with lactose and glucose, and we will end up with some transcription happening, since the repressor is not bound at the operator site, but it won't be a high level of expression since there is no cap to recruit polymerase. Therefore, we can say that we will have an activity level of 1.